Okay, welcome back to this second video of in pathology. As you know, the cell is the basic unit of life. So first, we're going to review injury at the cell level as a basis of pathology. Here you have the objectives we will cover with this lecture. You'll have to define and distinguish reversible from irreversible cell injury, explain the causes and the clinical manifestations that result, define and distinguish necrosis from apoptosis, and finally explain the different cell adaptations, what causes them, and what's the outcome. Cells need to be in a state of homeostasis or balance or equilibrium. And when that state is altered by adverse events, cell injury results. Cells have the ability to recover from mild reversible injuries if the insult, that's always a request, if the insult is removed. But some injuries are irreversible and changes to the nucleus or rupture of the plasma membrane occur. Injuries can be due to exogenous, or that means outside from the body, or endogenous causes, that's inside of the body causes, being the most important hypoxia or lack of oxygen. That oxygen is necessary for the normal activity of the cell, so if the cell doesn't have it, then it suffers some kind of injury. Lack of oxygen may be due to a reduced supply or an increased demand. When there is a total lack of oxygen, total depletion of oxygen, like in a heart attack, we call that anoxia. There are different situations that cause hypoxia or anoxia. For instance, when there is a reduced supply of oxygen at high altitudes. For instance, when you climb a mountain like the Everest, 8,000, almost 9,000 meters, there is no oxygen there. Or when the airways get blocked, for instance, when you are drowning. Also, if you have a lung disease that may prevent the blood to get oxygen, enough oxygen. There are many respiratory conditions like, for instance, asthma, which is quite common. Or also when the blood can't transport enough oxygen. For instance, in anemia. Anemia is the reduction in the number of red cells that you know are responsible to transport oxygen. Of course, when blood flow to the tissue is reduced to the, for instance, due to the failure of the heart as a pump. Also, at the cellular level, if cellular respiration is inhibited by a poison like cyanide, you know that respiration at the cellular level happens in mitochondria. And the lack of oxygen, hypoxia, results also in mitochondria not producing enough ATP. The cell will then swell, and if maintained for too long, the cell will die. If that doesn't happen, then the injury is reversible. Now think why a lack of ATP may cause the cell to swell. Okay, so the cell swells because water gets inside the cell. Okay, why that happen when there is no ATP? As you know, calcium concentration is high outside the cell and also in some compartments like the rough endoplasmic reticulum or the mitochondria. So if membranes are damaged, all that calcium can invade the cytosol, and calcium has the ability to activate different enzymes that will further damage the cell by affecting ATP storage, affecting the membranes and affecting the nucleus. All this is reversible to a certain point where the plasma membrane ends by breaking, and then the cell injury becomes irreversible. Irreversible cell damage leads to some clinical manifestations related to the specific function of that cell. For instance, if it is a heart cell, the heart will lose contraction strength. If it is a motor neuron, then the muscles won't get the proper input, and muscle paralysis may result. A case you probably know is when you damage your endocrine pancreas cells, which produce insulin. There is not enough insulin, so you become a diabetic. Cell injury can be detected by blood test because there are certain proteins that are only found inside a specific cells. So if you find them in the blood, you know there is some kind of damage to that organ. We call these proteins markers. For instance, there are cardiac markers like creatinine kinase, or liver markers like AST and LAT, or we also have LDH from destroyed erythrocytes. Okay, so when injury is irreversible, what happens next? Well, the next step is necrosis of those cells. And necrosis means localized death of a part of the body. There are different types of necrosis, as you see in the box, but the most common is coagulative necrosis, which is the one happening due to infarction or total deprivation of oxygen to an organ. Sometimes after necrosis, especially in the liver and the kidney, complete restitution is possible if the cells retain their ability to proliferate or to divide, in other words. If the remaining cells can't divide, that cell will be removed and replaced by fibrous tissue, which is not functional. This happens, for example, when you survive a heart attack. After recovery, part of your heart will be fibrous, and because it's fibrous, it can't contract, so the heart loses contraction strength. Sometimes, instead of fibrous tissue, calcification may occur. So you can find calcium, small calcium stones in your heart. In a stroke that affects the brain, as you see in the image, the dead cells are removed, but in the brain a fluid-filled pseudocyst results. All right. So what you see in this scan is pointed by the arrow is dark because it's water, it's liquid. On the other hand, apoptosis is a programmed cell death. This process may be led by membrane receptors in what is known as the extrinsic pathway, or it may be triggered by mitochondria, in which case we call it intrinsic mitochondrial pathway. Other types of damage can trigger apoptosis, like you are familiar with this, radiation, which is used in radiotherapy, of your or also your own immune system may cause apoptosis of some cells. Okay, and this image is for you to better distinguish necrosis from apoptosis. You can say that apoptosis is a clean type of cell death, while necrosis is more dirty type of death. And here you have a table showing the main differences. So you see that referring to the number of cells, apoptosis will affect just small groups of cells, while in necrosis are big quantities of cells. In apoptosis, the cell volume decreases slowly, while in necrosis what happens is that the cells swell and break. So the membrane is damaged, as you see in the box below, while in apoptosis the membrane doesn't break. It forms like bubbles in what we call retained in apoptotic bodies, right? So the cytoplasm breaks in bubbles. In necrosis, the cytoplasm is liberated into the extracellular space, and when you break a cell, what results is inflammation. You see it in the last box. So necrosis has inflammation, and apoptosis doesn't have inflammation. Referring to the nucleus, in apoptosis, chromatin condensates in what we call pyknosis, while in necrosis the nucleus breaks and the chromatin also. Okay, this map includes everything we just reviewed. It is made using the visual understanding environment from Tufts University. Some of you are familiar with it because you already use it. It is good that you become good and fast at making mind maps because all recent research finds that it is the best way to understand complex information and establish relationship between the different parts. All right, let's see now the incredible adaptability of the body and the cells. Cells, tissue, and organs can adapt to changes in different ways. Some adaptations are physiologic, meaning they are normal, and some are pathologic, which is not good. Some are reversible, which is good, and some are irreversible, which is sometimes bad. Okay, first one is atrophy. It is the reduction of the size of the cells of an organ or of a tissue. But a reduction of size is also possible by reducing the number of the cells in what we call involution, which is an irreversible process related to aging. When atrophy is pathologic, it may be due to disuse, denervation, lack of trophic hormones, those are the growth hormones, ischemia, which is lack of oxygen, or malnutrition, as you see this poor guy in the picture. Okay, the opposite is hypertrophy. So the organ increases in size due to its cells increasing in size while the number remains the same. This may occur only in the heart or skeletal muscle that you know from the last year that their cells can't divide. The heart hypertrophies due to several factors, but the most important is an increase of workload. Imagine you have hypertension for years. This means that the heart must pump against a higher resistance of the vessels, so it needs to adapt and become thicker and bigger in volume. Is that good in the long term? Well, no, because a thicker heart, as it's logical, will need an increased blood supply that depends in the heart itself. So you have a big heart that needs more oxygen, but there is less oxygen available, okay, so you have a problem. We will see this the end of this story when we get to the corresponding lecture in semester 2, which is cardiology. Hyperplasia means an increase of the total number of cells of an organ. Hyperplasia may be a natural adaptation and reversible, of course, like for instance when the uterus grows during pregnancy. okay, and then after delivery it regresses to normal size. In some other instances, it is pathologic, like in benign prostatic hyperplasia, which is really also a hypertrophy of the cells. So in this condition, you have hyperplasia and hypertrophy at the same time. Metaplasia is when a cell type is replaced by a different type of cell, and this is normally not good. For instance, the bronchial columnar epithelium may change to squamous metaplasia in smokers. So metaplasia is normally reversible if the irritant is removed. Otherwise, it may progress to dysplasia, and then it may progress as well to neoplasia. Neoplasia may be benign, a benign neoplasm, or a malignant neoplasm. Okay, we'll see that in the next lecture. So for now, this is all for cell injury and adaptation. See you in streaming in a minute.